Hello, my name is Spencer Fullam from Rush University Medical Center, and this is the video version of our poster presented at ORS 2022. This work was done together with Rahel Hunter, Dr. Anne-Marie Malfait, and Dr. Rachel Miller in the Department of Rheumatology. Calcium release activated calcium channels, or CRAC channels, open upon depletion of calcium stores in the endoplasmic reticulum, allowing large amounts of the cation to flow rapidly into the cell. At the plasma membrane, these channels are formed by the ORAI family of proteins like ORAI1, ORAI2, and ORAI3, which are widely expressed in many non-excitable cell types. ORAI is opened via binding to STIM on the ER surface, a process triggered by IP3-induced calcium release. Note that this pathway can be inhibited by the application of the small molecule BTP2 or promoted by the addition of the phytochemical thapsigargin. In human articular cartilage, one study reported differences in expression levels between OA and control tissues for CRAC channels and STIM. However, there exists little in the literature about the presence or function of these channels in articular chondrocytes and what the impact of rapid cytoplasmic calcium changes could have on gene expression and other cellular activity. So the goal of this study was to first investigate the expression of ORAI1 in murine chondrocytes, then explore its function through ex vivo calcium imaging using intact murine femoral condyles in control and modified mice with a partial knockout of the ORAI1 gene. Our experiments were divided into three efforts. First, a determination of the presence of these CRAC channels in cartilage tissues. Then, using mice with a genetically encoded calcium indicator, we looked to the impact of a CRAC channel inhibitor on intracellular calcium levels in chondrocytes. Lastly, we looked to the calcium response of chondrocytes with a partial knockout of ORAI1. We should note that all animal experiments were approved by the Rush Institutional Animal Care and Use Committees. For the qPCR test, femoral cartilage was collected from male wild-type mice at 30 weeks. Human cartilage was gathered from 6 ankle joints, made possible by the Gift of Hope Organ and Tissue Donation Network. In both cases, RNA was extracted using Trizol. Here, our ex vivo imaging system can be seen mounted on top of an inverted fluorescent microscope. For every imaging test, after the femur is dissected out, it is cut down, then glued onto a 3D printed holder to ensure repeatability. After the fixture is mounted, it is lowered down into a chamber of flowing buffered isotonic solution without making contact with the bottom cover slip. We change the solutions flowing through, removing or adding calcium or thapsigargin. Each image sequence was processed with the trainable Weka segmentation plugin for ImageJ. Delta F over F naught curves were generated for every frame of video for each cell. Comparisons between means of groups where P was less than 0.05 was considered statistically significant. For the qPCR test, we found naive mouse knee articular chondrocytes demonstrated high levels of the CRAC channel component ORAI1, which was expressed on the order of other known chondrocyte ion channels such as TRPV4 and piezo1. And for the human tissue, we found ORAI1, ORAI2, ORAI3, and STIM1 were also detected, with ORAI3 being the most abundant. For the first calcium imaging test, we compared the impact of thapsigargin to a vehicle control when extracellular calcium was taken away, then reintroduced to the flowing solution. Here we see a timeline of calcium levels in the solution and the presence of thapsigargin or vehicle control. We observed when the extracellular calcium was reintroduced, the chondrocytes that had been exposed to thapsigargin had a greater intracellular increase than the vehicle control. In a second experiment, pre-incubation with BTP2, a CRAC channel inhibitor, reduced the influx of calcium upon reintroduction of extracellular calcium. Lastly, for the second calcium imaging experiment, we compared an age-matched cohort with a modified mice line featuring a chondrocyte-specific partial knockout of ORAI1. We found that in the presence of thapsigargin, these ORAI1 knockout chondrocytes took longer than controls to see an increase in intracellular calcium when extracellular calcium was added back into solution. So to conclude, Chiefly, taken with previous findings on human chondrosarcoma cells, our results provide more evidence that CRAC channels are present and functional in articular chondrocytes. So little is known about the function or dysfunction of these channels specifically in chondrocytes that a closer examination could prove fruitful to better understand their role in ER stress. This pathway is better understood in other cell types, but understanding it in articular chondrocytes may help with the development of novel druggable targets to slow the progression of OA. Finally, I need to thank Cassandra Mercado for performing all the delicate femur extractions for the study, and I'd like to thank everyone in the joint Miller-Malfait lab at Rush for their collective help and collaboration. I'd like to thank the NIH for supporting our work through ORAI1 and P30 grants to Dr. Miller and Dr. Malfait. For citations, disclosures, and to learn more, please see this video's description. Thanks for watching.